Thank you very much for that question. That's a very, very important question in all of biomedical sciences, not only in dermatological sciences and hair pigmentation. Essentially, mice are nocturnal animals. They evolved essentially to be nocturnal animals, to be active during the night. So what is problematic for us in the pigmentation field is that much of human pigmentation responds to light. Our skin, for example, will tan under the influence of ultraviolet radiation. So it's rather complex to be using a nocturnal mammal to be the model of such a light responsive cell like the melanocyte. And the other thing to remember is that the mice do not really have melanocytes in their skin. They have melanocytes only in their fur. So that's another big difference. Now, if you move from mice in the wild to mice in the laboratory, one of the other big difficulties is that mice in the laboratory are inbred, whereas humans, of course, are outbred. So the genetics of inbred animals is really quite different from the genetics of outbred humans. And that's why humans are also different from each other, whereas mice are very similar to one another. And so we have to be very careful when we extrapolate results from the mouse, the inbred laboratory mouse, to a wider human population. The other thing to remember is that humans can grow long, long hair. Unfortunately, not me anymore, but most humans can grow long hair on their scalp, and mice grow very short hair on their backs. And the period of the hair growth cycle in humans, where hair is pigmented and hair grows, is called anagen 6. And this can last anywhere between 3 and 5 years on the human scalp. The equivalent subphase of the mouse hair growth cycle only lasts for 5 to 7 days. And so, if we try to understand the biology of what regulates the 5 to 7 days of mouse pigmentation for their hair, it's very hard to map that on to something like 3 to 5 years on the human scalp. So, there really, really are intrinsically different, underpinning fundamental biologies. The other thing to remember is that humans shed about 100 hairs, 150 hairs per day in the shower, for example. And that's because they have what we call mosaic hair growth, where each hair follicle does its own thing, is independent of the other follicle. Whereas in mice, they grow hair as a wave, where they molt a little bit like other mammals, where the hair sheds en masse, and then you have a summer coat, a winter coat, for example. So there's lots and lots of fundamental biological differences between the mouse and the human, and also between mouse pigmentation and human pigmentation. It's basically how do you predict the
Hmm.
data that you get in the mouse will be of value in the humans
Hmm.
when the scales are so different. Humans live for up to 100 years. Mice live for maybe 1.5 year. The hair grows for 3, 5 years on scalp in humans, but only 1 week on the back of the mouse. So many issues relating to scale are fundamentally different, as well as the genetics. And then, of course, the fact that the mouse hair grows as a wave, we call it synchronized, where every follicle is synchronized. Many, many thousands of follicles are synchronized with their neighbors. This makes it also very difficult to understand what is triggering these hair growth cycle coupled responses, particularly pigmentation. So another excellent question, and I think as we are finding in human medicine overall,
Hmm.
much of the data that we have discovered in mouse models has found it very difficult to translate to human models and even human medicine. And indeed, when drugs, for example, are released on the human market, many people do not respond to the same drugs that other humans do. So even within humans, there's huge variability of how they respond to the same intervention. So you can imagine going from mouse to humans is a huge jump if human to human is already a significant challenge. But I think there has been lots of discoveries in the mouse skin and hair follicle, hair cycle biology. We have discovered many genes associated with hair growth and with hair cycle, and many of those genes are also found in humans. That's not to say they all function in the same way in humans, even if they have in humans the same genes as in mouse in many cases. So there has been lots of basic biology discoveries of some of the more fundamental regulation of mammalian biology, but the more kind of intricate species-specific regulations are likely to be different. And one area that's likely to be very, very different is what we call epigenetics, the essentially the additional layer of control over gene expression above and beyond that which is encoded in the DNA. And we even see this between identical human twins. You can see identical human twins looking very different as they age in terms of their hair, their skin, and they're identical. They have the same DNA code, but their epigenetics may be different. So I think one of the areas that will be always fundamentally a challenge is to look at the epigenetic regulation for humans, that you will not necessarily find any clues in the mouse system. Thank you for that question. So if I take the first half, Um, I believe that lots of the new omics and other sophisticated tools that we have for understanding how genes interplay with proteins and how proteins then interplay with function can be tested in mouse tissue. If we're not expecting ultimate answers to the human scenario, but at least some clues because you can access mouse tissue much more easily than human tissue. You can go back again and again and again to the same genetically identical mouse to do spatial transcriptomics, for example, to map the skin, to map the hair follicle, to see what's happening to the melanocytes during the hair growth cycle. We know melanocytes in the hair follicle are tightly coupled to the hair growth cycle. And we can model the hair growth cycle much more easily in the mouse because thousands of hairs will be in the same stage of the hair cycle. So we can get perhaps more quickly some basic insights using especially spatial transcriptomics, where we see the location of the genes, the expression of the genes, specifically in which cells and where those cells physically are within the tissue throughout the hair growth cycle. And in the mouse, the hair growth cycle is so short, you can get your answers quite quickly. In the humans, it can take up to, like I said, 5 years. That's a long time to study any single human, 5 years. So the mouse will provide this kind of condensed time, restricted view of the hair growth cycle and hair pigmentation. That's a very important question, especially as we will spend a greater and greater proportion of our total lifetimes in this gray zone. So typically humans start to gray at around 30 years of age, and increasingly humans can now live to 90 and maybe 100 years of age. So you can imagine this is really the first time in history that humans will spend more time of their life, a higher percentage of their life with aging skin and aging hair or graying hair. So I think that there is a lot of interest to look at how health span is extended in humans, i.e. the function of our tissues, function of our organs, the function of our cells, for example, in the case of the melanocyte. So I think it'll be very interesting to see how we can maximize or extend the function of these cells, particularly in the hair. So you will notice that if people have black skin, they still have white hair when they grow old. So the melanocytes of the epidermis are able to continue making melanin for many, 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 many years, maybe up to 100 or more years. Whereas in the scalp hair follicles of these same individuals, or the beard follicles or other follicles, they already stop at around 30 to 40 years of age. So it'll be very, very interesting to see why does aging in the melanocyte of the epidermis, that gives us our skin color, appear to run differently than aging of the melanocytes in our hair follicle. And a lot of people would like to retain this appearance of youth for longer, and hopefully they'll be functionally healthier too. But if they could retain younger looking skin or younger looking hair, they may psychologically feel also when they look at themselves in the mirror that they are at least still more vital or more youthful. So I think it's important to understand how the aging clocks of melanocytes in different parts of the skin, the hair follicle, the versus the epidermis, how they differ, and to try and retain or slow down the aging, and whether that's through having better diet. In Western cultures, you have a lot of processed foods now, whereas before they would have eaten a lot of natural foods. And so we're not quite sure of the balance of nutrition that people in the West, busy people in the West, have routinely, and whether there's something we can do through exercise, through diet, through supplementation of diet, to reduce what we think are the drivers of hair graying, for example, like oxidative stress, or indeed potentially the drivers of hair loss, like smoking and other lifestyle interventions that may reduce our health span. Think there's a multi pronged approach. I don't think
Yes.
any single strategy. I think individual strategies may contribute a little, but together they may either synergize or they may accumulate to be a more meaningful intervention if they're done together and in balance.